This video was produced by
Welcome, Grace.
Okay, thank you. And the reason I'm talking today is because I need help, okay? I need some input from other disciplines, particularly people from the artistic disciplines, to help me use persuasion to grant applications for reviewers, just when I want to explore a new idea, a novel idea that some people may consider wild or crazy. And in my career, I've done such things and certainly met with opposition. Eventually I succeed, but it takes a long time. And I want to find a shorter way to persuade people to dispel some of their biases, open up their mind, and think about a problem in a new way. Now, I'm a scientist, and I used to think that, well, scientists, we look at data, and we look at what the data tell us, and we can change our ideas and thinking fairly quickly because most of us are objective. Well, maybe many of us are objective, but I've been astounded by how many are not particularly objective. And there's a long history of this. For example, there was a woman named Barbara McClintock. When she was in her 30s, she started working on corn, and she was the one who discovered that there are genes that jump around the genome. They're called jumping genes. And people thought she was crazy. They used that word. And fortunately, she was wealthy, didn't have to have a job, so just worked, didn't need to be paid. So she just worked for about 50 years on this problem, and 50 years later, just before she dies, they give her a Nobel Prize. And so she sort of broke the record of how long before she made the discovery and tried to explain it versus people recognizing it. There are other issues. Another scientist, any of you who've taken general biology, you learn about the hypothesis, or now we call it a theory, of how the eukaryotic cell evolved. The eukaryotic cell is the cell in every plant or animal or single-celled protozoan. And it's basically when two bacterial cells got together in a symbiosis that led to the evolution of the eukaryotic cell. Well, Lynn Margulis, a professor at University of Massachusetts in Amherst, she died 2 years ago, but in the late 70s or 60s and 70s, she came up with this hypothesis. It took her 17 different journals submitting a paper and getting a rejection, going back to the next journal. 16 journals passed on this. The 17th accepted it. This theory is now in every single biology textbook and widely believed. And it took quite a while. And I'm not saying this is a gender thing. Think about Galileo. 1600s, he was threatened with beheading by the Church of England because he said that he countered the idea about the Earth not, people used to think Earth's center of the universe. He said, no, it's not the center of the universe. And it wasn't until the 1980s that the Catholic Church actually published a statement, yes, we agree with Galileo. That broke Barbara McClintock's record, you know. So anyway, there are many instances. And what so basically what I'm interested in is what happens, or how do you present an idea that's novel, that breaks the rules, goes against people's thinking. Okay, I have found that data alone are not sufficient. Okay, a variety of people present the data. Okay, and one obstacle is that sometimes if a problem is very complex and it has a lot of parts that are strange, that maybe it obscures the germ of that idea that you're trying to get across. So, what are some of the solutions? Hi, come on in. Okay. No. So one of the solutions, do we provide all of the data, and do we tell the story as we sort of understand it or know it? Okay. I can tell you right now that hasn't worked very well for me, okay? Do we emphasize only some of the aspects, maybe the parts we think people can handle, you know, and de-emphasize other aspects? To me, sometimes that seems a little bit intellectually dishonest, okay? And you can go even further and really simplify the study and omit important parts of the story that you think maybe people aren't ready for yet, can't handle. Well, the one thing I do know for the solution is we need to illustrate, okay? But how do we do that? There's a long history of biological illustration, but that's usually trying to give the most accurate, realistic image of a morphological structure. But here I'm talking about ideas, processes, and phenomenon, okay? So I'll tell you about one phenomenon I continue to work on, but something that started I started working on maybe about 15, 20 years ago. We've got a general rule, sort of a dogma in biology, that all of our cells in our body, so you could use the soma, but just think in our body, that make up our skin, our intestine, nerves, heart. In the human, it's been measured that it's got 7 gigabases of DNA. So that's just an amount of DNA. And we know that the germ cells, when mom and dad got together and produced us, mom contributed 3.5 gigabases, in other words, half our chromosomes. Dad, with his sperm, contributed the other half. And in almost all organisms, plants and animals, the germ cells have half the amount of DNA as that progeny that comes out of it, the adult. And that way our genome size doesn't continue to get larger and larger. It keeps this sort of stasis. This is the general rule. Now I'd like to introduce this arthropod. It's a crustacean, sort of shrimp-like, but small, 1 or 2 millimeters. It's called a copepod. And this is a female, and she's got her body, or soma cells and an intestine, and she very conveniently carries her embryos and sacs outside her body. And this particular species breaks the rules, because if you look at the DNA in the soma, the intestine, the antennae, it's got 3 gigabases. If you look at the germ cells, 15 gigabases, okay? And this species is not the worst breaker of the rules. There's a species I work with in Russia in which the germ cells are 75 times larger than the soma, okay? And I won't go too much into what this DNA is, but we know it's highly repetitive. It used to be called junk DNA. And they're actually largely genetic parasites. So anyway, they break the rules by having obese germ cells. So try and think of this obese sperm trying to swim and get there first to the egg. People say, why would they do that, you know? Okay, so, sorry, okay. So I'll tell you about how they accomplish having a very large germ line and a small somatic line. This scheme shows the divisions of the cells during early embryogenesis. And all of you might not remember, but you all went through this at one time. You were a zygote, okay?
Yes.
Remember being a zygote, okay? Okay. And then you came the 2-cell stage and the 4-cell stage. You've gone far beyond that by now, 8-cell stage. But in these copepods, or in some species, when they get to the 16-cell stage, 16-cell stage, 15 of the 16 cells chop up their chromosomes, throw out 80% of their DNA, glue their chromosomes back together, and go on with life. And they form the stem cells of the somatic line, the antennae, the intestine, things like that. Only one cell is not diminuted in its amount of DNA. One cell, supposedly, that's what the model says, remains unaltered. So this particular phenomenon was shown in a study published in 1977, and the woman who did it, Sigfrid Biermann, took 17 years to do her dissertation. That's another story. But it took a while, okay? But she managed after 17 years to get sort of the story together. But in her story, she had only a small amount of DNA thrown out. So people recognized, yeah, it's an interesting phenomenon, it occurs, but in her situation they throw out only a gigabase. Okay. So I start to study this phenomenon.
Was that notion of throwing out DNA, was that a completely new thing? Like, did that not happen anywhere else? And so when she said that, everyone...
Okay. It's known to happen in ciliates, but in ciliates, single-celled protozoans, they throw out little pieces in just a small amount. It was discovered in the late 1800s by Boveri, who worked on parasitic nematodes. Ascaris boscaris, they parasitize a lot of horses, large mammals. So it was known there. When it was first described in copepods, it was described as aberrant. These are sick, you know, damaged or whatever. So Bierman is the first person who showed, no, this is a normal part of their process. But I won't go into the details, but she sort of measured the wrong cells or figured out, she didn't quite measure how to figure out what was being thrown out, okay? And so I started collaborating with, well, I don't know if I should tell a story how I got interested in this. I was a graduate student and got a letter from an editor of a book. It was a book on developmental biology. And he knew about this system, and he was trying to encourage developmental biologists to consider organisms like this in studying development, and not just the chicken and the frog and the mouse. And the reason he contacted me is Sigfrid Bierman, after working 17 years on her thesis, came down with breast cancer, was not able to write. And he knew, somehow, I have no idea, but somehow he knew that I worked on copepods, I could culture them, I knew the methods, so he said, would you write a chapter on the methods and review the literature? And so, being excited and a naive graduate student, I said, yes, of course. And I think I hung up the phone and go, what's chromatin diminution, you know? But then I certainly found out. I reviewed the field, which didn't take very long because there were many papers, you know. So anyway, so then, but that was in the early 1980s. PCR, polymerase chain reaction, that amplifies DNA and enables you to study the DNA, had not yet been invented. Next generation sequencing hadn't been invented. The costs for next generation sequencing had not yet come down. So I just started studying the cytology and squashing them in their chromosomes, but I didn't start the molecular work or measuring for some time. And then I get to JMU and I decide, there's mesocyclops, there's this copepod right in Lake Shenandoah, and I think I'm going to pick this up and just start studying this phenomenon and wait for the molecular tools to develop. And I form a collaboration with a professor at the medical school at East Tennessee State, Dr. Ellen Rash, and she is arguably the best quantitative cytogeneticist in the world. She's credited with having the most accurate estimates of amounts of DNA when measuring them in various organisms. And she's the one that taught me a lot of techniques, and she and I collaborated. And so we started looking at the amount of DNA, and we thought, oh, only a gigabase is going to be thrown out. Well, we found out that it was 15 in this and a lot larger in others. Okay. And this is sort of the story that we have an idea about, but there are lots of other strange parts. For example, we think that there are additional diminutions in the sperm, that the sperm only contributes 3 picograms. We don't know how much the egg cell. So mother and father contribute unequal amounts. And then somehow the chromosomes get together. Now the mother and father chromosomes are physically separated, don't even commingle until after the diminution. That's sort of weird. Then finally they throw out some DNA. But we started to measure the amount of DNA in the germ cells, and the amount of DNA in the germ cell, which we think should be 15 picograms, changes during development. So there's an additional phenomenon called endoreduplication or endocycles, where maybe the DNA can duplicate but not separate into new chromosomes. I don't want to get too much into the biological details except to show that our first approach was to show all the measurements that we've made and at the various stages. Because before, Bierman just measured an adult female and a sperm, and that was it. And so when we presented this, something like this, in a story form to a grant proposal to the National Science Foundation, and I've worked at the Science Foundation, I know how to write proposals, I've had many proposals funded. Well, when we submitted this idea, went to the eukaryotic genetics panel, and I've read thousands of reviews having been at NSF, and these were among the worst reviews I'd ever read. One of the reviews said they named famous geneticists that would be rolling over in their grave if they read what Ellen and I had written. They said Wyngaard should be kept out of the classroom. She will just confuse students. And the program director said this was the lowest ranked proposal in the competition, you know. So what do Ellen and I do? We know we're right. So we just go back and you just collect more data. And we did that, and eventually we got funded by the National Science Foundation to do some work. And then with Brian Walton, we just got a big NIH grant to pursue this. But it took 15 years, okay? And how do we get people to entertain different ideas and think that we're not crazy? Well, maybe I need to simplify things. So here's my when I look at a copepod, I know you were bowled over by their aesthetic beauty and whatever, and their colors and stuff. But when yes, but when I look at them, I see the amount of DNA in their cells. And so, this pink is a stain that's quantitative. So in our lab, we take the stain and we stain the cells, and the DNA is bright pink, and you can measure it, you know. So, and I see that in the somatic tissues around the intestine and eye and antennae, that's 3 gigabases. And then we measure the amount of DNA in those embryos, you know, from the zygote on, before the diminution, and that's 15, okay? And then, but we also measured germ cells that were undergoing maturation, their process of development, and they should either be 15 or 30. 30 for when everything doubles and you go through 2 meiotic divisions. But we get this, some of them, we get a whole range that isn't quite right. And so people don't understand this, okay? And I've left out the part that the sperm is only 3 picograms, okay? Oh no. Okay. Well.
Can I ask a question?
Yes.
Well,
yeah. Go ahead. No, ask. No, ask.
So, is the problem that I guess my big question was the question of audience.
Right.
Because the big question would be, how much do the people who are reading this know?
Mm-hmm.
So if it's and it sounds like they know a lot.
They're very intelligent people.
But this is also not a general board.
Right.
This is a board of specialists in your particular field. So this is not, let's dumb this down, make it easier, more accessible.
Right.
It's a question of how do you communicate best to people who do pretty much exactly what you do.
Yes.
Okay.
Yes. Right. And the next figure, I'll just quickly sketch it. And it's a shame because I don't know why it's not showed up on my computer. But it's got the copepod, okay? And this was actually worked on by Jonathan Smith, who took a biological illustration course last year with Alex Bennigan. And we simplified it. And so they have their eggs. And all we said is they've got 15 gigabases of DNA in their eggs. They've got 3 gigabases. But what he did was he sort of drew a cone, and he drew a big nucleus right here. And then he had sort of a cone here, and he drew a little nucleus. So basically, we used a model. So when we applied for the NIH grant, which got funded, And when I went to meetings in Santa Fe, mobile genetic meetings, in which I was the only one who worked on a copepod. Everybody worked on nematodes, yeast, mouse, frog, and I had to introduce them to what a copepod was. And I presented it very simply. All I told them was the soma's small, the zygote's big. It was very well received. Brian and I got our NIH grant funded. So when I sort of dumbed it down or just told a little bit of the story, very well received. But I left out the part about the sperm, about during juvenile development. A lot of other things are going on. Well, what I wanted to point out was the student, Jonathan Smith, he took my diagram and he started just doing little annotations, like a little cone that goes out and tells a little story. This January, the issue of Science, which is regarded by many as the premier scientific journal internationally, had a competition on visualization. And the one that won for the year 2013, presented in January, it was a master's student working on hypothermia in sea turtles. And sea turtles, in the winter, they may go down south, okay? But sometimes they get into water that's very cold and they undergo hypothermia and they end up on the beach. And I'm not going to go through all the details, but what she did is she made a poster to put on many beach communities so people could walk on the beach and recognize, is the turtle okay, or does it have hypothermia? What contact, who should I contact to help me with the turtle? And so and it affects various parts. You know, there are parasites and problems with the stomach and lesions. And so she sort of had these little cones where she'd tell a little story. And so I told John, look, when you started your little cones of telling a little story, that strategy sort of won somebody a competition. So maybe, you know, he was onto something. But basically, what I sort of want to ask you for is feedback on how can I use visualization in art for the next set of studies, like why or how is it that the dad contributes a lot less DNA to the zygote than the mother, you know? And the other idea I have, but it seems people aren't quite as reluctant to believe this, is what is this DNA? They used to think this DNA was just what we call junk DNA, repeats, cytosine, cytosine, adenine, cytosine, cytosine, adenine, or something like that. They're actually genetic parasites. Those are sequences of DNA that invade the genome and they get the host to copy themselves. And even presenting this idea, people said, but how can an organism tolerate such a high parasitic load? And I have some ideas for that. I don't think it's quite the place now for me to go over those. But what I want to say is I have some more wild ideas. I don't have 15 years, I don't think, before I want to see them go to fruition. And I have found simplifying an illustration in the last year or two to be very productive and financially rewarding, okay? But I need to go further, and I need to go beyond what is useful in many regards. Many of you may be familiar with the work of Edward Tufte, professor at Yale. He has a variety of books. One of them is Visual Display of Quantitative Information. Ellen Rash, I said she was a quantitative cytogeneticist. She and I build models. We measure things, and people still argue with the numbers. We submit a paper, and they don't believe it's 75 gigabases or 15. And, you know, we're reporting the data as we see it. So, and then Tufte tends to present things very accurately. It's my impression, very realistic and accurately. And that's something I like as a scientist, to be honest. But somehow it's not meshing with the artistic approaches that get across. So I will stop there and ask you for your input.
I have a question
Yes.
of the copepod.
Yeah.
Is that, from what I can tell, it's completely unique. There's no other thing that you can really compare it to in the way that it functions.
You mean as far as its general biology?
Yeah.
Yeah, there's nothing morphological that you can see. It doesn't necessarily mature faster. It doesn't have a larger body size or a smaller body size.
Okay.
Yeah, I will note that the group I work on, only a couple of species do it. And they tend to be subtropical and tropical in origin. And they can withstand temperatures up to 35 degrees centigrade, which would kill most copepods. Well, this kind of DNA, this genetic parasite, thrives under stress. So maybe that's one instance. But I have another hypothesis. So that's one kind of stress, thermal stress. And that has allowed these parasites to proliferate because the host defense system has been weakened under maybe heat stress. Well, if you go to some alpine lakes in Europe, very high altitude lakes, and some of these lakes have so little nitrogen and so little phosphorus that the only thing in the lake is fish and these copepods. They don't even have like Daphnia, the water fleas, which are very abundant, because hardly anything can live at such low nitrogen and low phosphorus. How is it that these cyclops, these copepods, can do it? Well, my idea is that, and it also takes them, instead of just taking them 2 weeks to develop, it takes them much of the year. Well, we all know in ecology, from ecological studies, that what mothers often do is they sequester yolk in food for their young. So when that embryo is being formed, it has lots of yolk to continue through development, and then maybe even a little bit of yolk left over before it goes into a larval form. Well, I think this DNA that's being dumped out is what sort of like a yolk genome. Because if you are living in this lake with hardly any nitrogen and phosphorus, and these are things that you need to make DNA, to replicate your DNA. Well, I think the mother's storing up all of these nucleotides, all of this DNA, and so she puts it into her embryo, and then after a few divisions, it's going to need some nitrogen and phosphorus, all those little things you know that makes that double helix. Well, that's dumped into the nucleoplasm and broken up, and so it'll take it through the next few divisions. Otherwise, it'll never make it out of the egg and into a larvae. So that's sort of the idea I have now. And I presented that last week, and people actually liked it. I was a little concerned or worried, but they actually liked it. So,
how did you present it? Just verbally and with?
Yeah, verbally, but I used the pictures of the copepod, and I talked a little more about the biology, and I used a picture that fortunately is not showing up. Showed up on my Mac, but it's not showing up here. Yeah.
And are they beyond photography or photomicroscopy? Are they so tiny?
Oh no, we take pictures of all the cells all the time, and then the whole animal itself just with the dissecting scope.
But those were drawings from it? Or?
Drawings, yes, these are drawings. Certainly I can take photographs, but you'll just see what everybody else sees, a beautiful animal with antennae and nice eggs, you know. So, you know, so I'm sort of, I think
the diagram needs to distill out
the information.
Like you were saying,
Yes, and not watercolors or drawings. I think computer illustrations, because Jonathan Smith, I said, I want you to, he says, oh, I'll draw it. I said, no, I want it in Adobe Illustrator, because every semester my students are going to make some progress and we're going to change it. And so, and Jonathan's fine with that. We start with something, and then as we understand something a little better, we can manipulate it. Yeah, Ralph.
Well, following up on that change in the kind of sidewalk conversation we
Yeah.
had yesterday evening, seems to me there was an old paradigm for this.
Yes.
On the relative proportion of somatic to germ
Yes.
DNA.
Right.
And now there's sort of a new paradigm
Right.
in terms of that ratio.
Right.
Have you used, you know, and it's sort of like the paradigm shift that you see in the structure of scientific revolutions.
Right, yes.
Have you used an image that conceptually represents the old paradigm alongside an image that conceptually represents the new paradigm, so that people can kind of, like if you had these two screens here,
Yes.
this is the conventional
Right.
wisdom,
You know,
this is a new approach.
well, I hope to write a book and do that, but I tried that in a practice seminar. I was giving a hour-long seminar for our department, and so I had the old and the new. And one of my colleagues, whom I have a high regard for, said, Grace, just take away the old. It's going to confuse people. Don't tell people how you got there. You don't have to tell them the whole story. But I actually, so I said, okay, I'll leave it out. But I really think to fully understand it, and I think I'm only going to be allowed to do that, because it's not passing through review or whatever, is when I write my own book, you know? And then I'll go, yes, this was the old paradigm and this is the new. Yes, I like that, and I'm glad to hear that you do too.
Yeah, and kind of in keeping with comparison and what David was saying about audience, I was thinking about, you know, when Darwin first presented evolution. And
that was another example.
Pigeon, breeding, which was something that everyone could relate to, and then you had something that you could show, like if
yes.
new and a new,
yes.
even, or new and old, you would still have that same concept working in
right.
the brain.
And I think I might show them I'm not throwing everything out.
Right.
We're not breaking all the rules, just some of them. Yeah. Yeah.
I'm not sure without seeing actually the data to know how applicable this would be, but have you thought about looking at, so you've run a statistical quantification of like the gigabases and the different types
yes.
of lines. Could you use data visualization softwares to graphically, you know, visually represent those differences?
As they occur during development? I've thought a little bit about that. I don't have the expertise. Somebody who I think would be great at that would be David Bernstein. And he emailed me, and he's got another meeting, wasn't able to come. But I think, and we see some visualizations in a lot of our textbooks, you know, they show DNA replication, they have these boxes come in and do this. I didn't want to quite do that, but that's my initial approach, to show something quantitative and dynamical. Of course, right now I'll have broken parts because I don't understand every part, but
I think a lot of the data visualization work is becoming more encouraged in scientific communication. I know in Harvard they just did a workshop entirely on data visualization in the, I think it was mostly astronomy and physical sciences.
Right.
You could easily translate.
Yeah.
And I think they're putting those visualization products forward as communication methods, because I think a lot of people are just, they want the data, as you know, they want to see the data, they want to share the data, but it's also dry.
Yeah.
And this is a way to reach another audience.
Yeah, because on the very detailed diagram, I wanted to put it in a grant proposal, and one of my collaborators said, no, they won't even read it, you know. And I remember you telling me when I was talking about a book, David Ehrenpreis was saying, Grace, you know, you got to get modern, you know, you got to get some animations, you know, maybe you ought to do a course with animations. And that may be one way to get some of this across. And of course I said, well, I'm not opposed to it, but I don't know how to even start that. You know, I would need a collaborator.
I thought about this too, because part of it, I mean, this balance between aesthetics and information is really fascinating. And of course, Tufte is all about that.
Yes.
And I think the ideal there is that somehow the absolute perfect still point is equal parts information and aesthetics. But and God knows whether one can actually achieve that, because this is all about sort of having to lie in some way.
Yes.
It reminds me of the conversation that I had with a friend of mine who there's a woman at MIT that you all, I'm sure, have probably heard of, named Felice Frankel, who does all of the, when you see a cover for Science, she shot it.
Oh, I see.
Right? She does the incredibly beautiful data visualization, scientific data visualization.
Okay.
And I remember having this conversation, talking about it because I'd seen some beautiful image, and my friend the scientist said, yeah, oh, that Felice Frankel, you know, yeah. And was just horrified by Felice Frankel because she was lying. Like, she, you know, it was not, it was not objective enough.
Right.
And she was,
I struggle with that.
With things and making them look prettier than they really were, But I will say that, I mean, one thing that comes to mind is like, you know, making something stunningly beautiful never hurts. I mean,
Yeah, that's that false color photography for all.
Yes.
all
Right.
astronomy images, you know,
Yeah.
don't actually look like that.
Right.
It's like heartbreaking to find out as a kid, right?
Really? I just found out now. Okay. Okay.
Well, I told you about Brian Augustine, who's no longer here, but Brian
Yeah.
Augustine did nanotechnology.
Yeah.
I remember going to visit him in his office, and I'm like, we're chatting, and I look on the wall, and he's got a calendar. And I say, oh, that's interesting. What is it? And it's a calendar that's sponsored by, you know, like some
Nikon?
company.
It's the Nikon calendar?
And I say, oh, that's really neat. He says, no, yeah, last year I got an honorable mention. And so, you know, here's
Yeah.
the scientist.
Yes.
Like very carefully submitting these images, not because they're, I mean, but because they're beautiful. And
Right.
you have this whole wonderful pinup of these beautiful scientific images. And so I don't know how that plays into your question in terms of the people
Yeah.
on the panel, and I don't know whether that actually helps or hinders, but in the in the larger audience, it kills. Like, I mean, it will.
You started out by saying, what's the appropriate percentage of art and data and accuracy? Maybe does it change with the problem? You know?
I think so. This idea that you went to with Darwin of rooting it in a known
Yes.
culture, right?
Right.
And then presenting this new idea in this known framework is really valuable. And if you know your grant is being evaluated among the
Right.
hundreds
Yeah.
of other grants in that same classification, and it all becomes another idea of how to increase your competitive.
Right.
And if other institutions are employing illustration or data
Yes.
visualization to tell this story, which is what you're doing,
Right.
telling the scientific story, and then you're presenting a tabular data or something like that, it's not as compelling or.
Yes. Because I did it in words in that grant proposal that was very poorly received. I did compare the old and the new, but it was done in words, not with images. And I might have made a mistake there. Yeah.
Following up on this idea of lying,
Yes.
I know clearly if you take mice and you use a marker pen to draw lesions on them,
Yes, that's bad.
that's falsifying data.
Yes, right, yeah.
In this case, though, are we really talking about excluding certain data to come up with a much simpler and more easily understandable conceptual,
Yeah.
representation of
Yeah.
the idea, versus actually taking the data and falsifying them to.
But is it
You're not
lies by
actually
omission?
tweaking the data, you're
Right.
just excluding certain
Yes.
data to make it. But don't we always do that to separate signal from noise in the world? Don't we always
Well, that
exclude all of this constant data that's
That
flowing
inside-
in?
But it's not noise.
Yeah.
It's not. And the thing is, I read one quote where somebody said that in science, or like biology, we tell a lot of lies. Because we think some students don't like biology because in high school or K through 12, they thought it was all facts. And facts are like false or true. Much of biology ends up going through revision. And so a lot of what I'm teaching, it's maybe close to the truth, as close as what I know to be the truth, but it's going to be revised and changed. As we learn more, our lies get smaller and smaller. So you can look at it that way. But I am afraid about lying through omission. You know, when I know it's far more complicated than that. And I purposefully left out some of the parts that I know people would just struggle with.
But
Yeah.
I think you could argue that purposes of a grant proposal, let's say, there are aspects of the data that you want to highlight to tell the story with the intention that a publication or something will come out of it that will have all of the data analyzed and be a more complete picture, but you need to get that funding to.
Yes, right, yes,
You know.
yes. Yeah.
So I think since that isn't the main form of communication to the community.
Yeah. Yeah, I could see that. Yeah. Although we've struggled when we've had this paper, the one with the pink diagram, when we sent that out for review, there was one reviewer who just argued about the numbers. No, your numbers are wrong, even though we showed the data and even tried to show that we calculated it wrong. And then others just say, well, parts of this, if you present something that's crazy, like, you mean the mother and father contribute unequal amounts of DNA? They see that in a publication and it casts doubt on your other stuff. And so you run into that. It's like, well, if they think that's going on, which is really crazy and has not ever been reported before, how do I believe this other stuff? You know. And again, I don't want to wait another 15 years. You know. So.
The other person that makes me think of is Chris Rose, because I mean, I remember having this conversation with who also does biology and does morphological changes and stuff like.
And Chris is a deep thinker.
But I remember him bringing in, we had an exhibition a long time ago that had his, it was about changing methods of scientific illustration. And he had 5 different ways he had represented these creatures, from the hand-drawn version to the it's on the computer to the, okay, how am I going to stain it? And a lot of the discussion was with the staining and how did you stain best to demonstrate morphological change on specific organs? And it was quite clear, like, that's not about being objective or not. Whether you decide you're going to do that in red or blue, like, I mean, you can, and that's an aesthetic decision. So how you actually demonstrate those changes can totally change the way people perceive it, and is aesthetic, and isn't lying in any way. And so, and it's not objective. So I'm just thinking, like, in the same way, all of those lessons seem applicable in this instance, that they're going to be different ways, and some are going to be better than others. And it's not going to be because one's lying or leaving out stuff out. It's because they're, I mean, from
bringing certain things to the forefront.
When I think of Tufte, and when I think about sort of visualization, which is a word which I hate, but like creating meaningful, persuasive pictures, the ones that are most effective, just like good writing, are the ones that are the simplest and the clearest. And so, if it's complicated, whatever it is, if it's the grant that's written that way, if it's the illustration, if it's the diagram with 19 moving parts, it's not that people are stupid and just can't take in that information. It means that the information could be conveyed in a clearer, better way. It's like when I had to write the abstract for my dissertation. You know, you've like written 300 pages on your dissertation, and UMI says, okay, write 150 words. And you're like, and then you write 150 words. And guess what? There are 150 really good words. So it's the same way. I mean, the problem is, okay, show it to me like this. And so that, and I think that happens with elegance.
And when you're saying, how can we use art, I guess, like as a photographer, I take that for granted. Like some things maybe should be in focus and others left out. So viewers looking at this, not that in the background. Or the angle is such that I'm showing you this, I'm not showing you that. It's all there. I'm not saying it's not all there.
That reminds me, the first time we submitted the grant proposal, we used a photograph of this distinctive cell that says the animal has diminution. And it's this big, fat cell with little veils of what we think is the euchromatin. Very strange morphology. In fact, it made the cover of the Journal of Morphology when we published it. So I thought, hey, great photograph. And it made the cover of the Journal of Morphology. You can't get too much better because it's, you know, morphology. So let's put that picture in the grant proposal. Well, one of the reasons the first attempt was not funded, they said, you really need state-of-the-art photography because your nuclei look really strange, you know? Even though I had explained it had a distinctive morphology, but they looked at it and go, doesn't look like it should look. Therefore, your photography must be bad. And when it was really capturing reality.
Right.
You know.
When I'm thinking like with a comparison would be with photojournalism, it's supposedly neutral, but it's actually so not, because it's the same thing of like, what are they showing and what's over here that we're not seeing?
Right.
What's emphasized. It's almost like, to me, it almost is unavoidable. And I know like with science, it's a little, it's kind of maybe like photojournalism. There's like an ethical responsibility.
Yes, and I want to keep.
Even with that, you know, it's fine.
Because as a scientist, if you do something that is even perceived as damaging your integrity, it's over. You know, you cannot lie, you cannot be sloppy, and once you're sloppy or you commit fraud, people will not trust you again. So maybe that's sort of in back of my mind. I want to keep that integrity.
I understand
and we're about,
like,
yes, time to go. No, it really is time to go.
The idea.
Thank you very much for coming, Ralph.
The idea that there's a difference between a photojournalist who knows in his heart that the Vietnam War is insane and takes a picture of someone executing someone else with a gun.
Yeah.
And like, it's pretty clear you see that shot in an anti-war picture.
Yeah, right.
Right? Like, it's an anti-war picture. He believes passionately that this is something that is awful. And he wants to show it in a persuasive way.
Yeah.
You believe something.
Yeah, I believe my model.
True, and you believe it is the way it is, and you want to convince people that this is the way it is. And I don't see that it's any different. I see that that is precisely, it's a question of, and the works that stick are the works like that picture, that all you have to do is look at that picture and go, I have nothing to say. Like, there is no argument. It's that right there. And I understand that it's different with science, but I don't think it is different. I think in general, when these leaps are made, people eventually just, right, that, yes.
Right.
So that's not.
You're not so sure? Yeah. Yasmin.
You're saying it changes over time. You want to be able to change it as you're learning more.
Right,
and,
yes.
You know, different things come into the gaps in science that you want to fill in, and it's hard to find that balance there between truth and then living yourself.
But at this meeting in Santa Fe last week, all top biggies from all around the world. In fact, I was the only one from a primarily undergraduate institution. I provide the simple one, and they loved it. But if I had told them about the sperm being really small, I don't know, that would have opened up a can of worms. You know, it was the first time they were being introduced to this at all.
That's the other thing, is that if it's so groundbreaking, right, so alien.
Yeah, alien.
And that it has to be constantly couched in familiarity to get.
And I think that is, and that comes back to the comparison, couching it in familiarity. So it's not like I've got an alien brain and I've got these alien ideas.
Well, and even with Darwin, it took forever, you know, I mean, still is an issue. And, you know, we're constantly trying to find new ways. I don't know if you've watched the new Cosmos that's out, but they're trying to do things with animation and relate it to dogs because so many people have relationship with dogs. And I mean, I think even Darwin used dogs.
And Darwin took 20 years to synthesize, but of course, he lacked the courage for a long time to come out with the ideas until his hand was forced by Wallace, you know, so.
And I also don't know that we can, I think it could be troublesome to say if you feel passionately about something in science, then you just focus on this area that, so you can tell your story. Because just because you, like, you shouldn't, science, you shouldn't be feeling too much about.
That's what they say.
You get into a lot of trouble feeling ways.
You know? Not even supposed to use the word. You say, I believe.
Let me ask this question.
Let me be devil's advocate, because
does she care about Google Bonds?
of course.
I'm passionate.
You have passion.
More than I do?
You can have passion for your research topic.
Yeah.
And you can present the evidence that you have, you know, and convey your passion in various ways for the evidence that's there.
Right.
But it's when you speak on the passion and don't support it with the evidence, that you open the door for things like MMR causes autism. Honestly, you know, people feel very passionately. It was totally bunk evidence,
right, but
the passion overrode the science.
All I would say there is that the picture in the example that I gave is the evidence, and the picture is incontrovertible.
But you can't make an analogy between the photojournalism of something like war, I don't think, of something like war to the explanation of scientific evidence. I don't think the analogy, while it may work for you, it doesn't work for me.
That same photographer, I know the iconic one you mean, may have other photos that don't suggest it's harmful. Like, you don't know that.
Yeah, but that's the one.
That's just one piece of the data.
The boring ones that look like a normal day in the life of the war.
That's the one that became evidence.
Yeah, but that
in other words, it became evidence because it rang true. There are lots of other pictures.
That's
the reason
my point.
that
There are
So
lots
it's cherry picking.
It's
Cherry picking the data.
And I don't know that the photographer did that. I think just culturally that bubbled forth as iconic.
Thank you very much. Okay. Okay. Thank you. Okay.